This is just like a summary of what I did that year, that summer, and it dealt with obesity in the hypothalamus. We were working with a colony of mice, and we knocked out some genes to see what were the effects of that in the metabolism of some mice. This is a very basic overview of what I did that summer. So we focus on the hypothalamus, and it's essentially, well, I'll explain it, and its relation to obesity and diabetes. As you know, obesity and diabetes are closely linked. Hypothalamus is a region in the brain, it's about the size of an almond, like right above the brainstem. And basically, it's responsible for sleep-wake patterns, temperature regulation in the body, and feeding behavior. So we're focusing on the feeding behavior part. So this is just an overview of the nervous system. It can be divided into the central nervous system and the peripheral nervous system. And this deals with the brain and spinal cord. And the peripheral nervous system is, as the name suggests, dealing with the peripheries of the body other than the brain and spinal cord. So like your nerves and your things, you know. So it can be divided into somatic peripheral nervous system and the autonomic peripheral nervous system. Somatic just means it deals with voluntary behavior. If you pick up a ball, that's dealing with the somatic part. The autonomic part, which is what we were dealing with, can be further divided into sympathetic systems, and the parasympathetic system. Sympathetic can be summarized as the fight and flight response, and the parasympathetic rest and digest. So if your heart is accelerating, if you're scared, you're dealing with this system. If you're breathing, rest and digest. So the hypothalamus, there are basically two main sides. The lateral hypothalamus is responsible mainly by making you feel hungry, in essence. And the ventromedial part is responsible to making you feel full. This is research, I believe, from the 50s. I forgot the name of the scientist who focused on this, who discovered this part. Starts with the F. I want to say Friedman, maybe. I'll look that up for you guys. But in this one, in this subject, the ventromedial hypothalamus part was knocked out. So it never, the lateral side of the hypothalamus was burned. So it never felt hungry. And in this one, the ventromedial side of the hypothalamus was burnt. So that center didn't work for this mouse, and it never felt full. So that's why it was like that. Some of our subjects were this size. So what we did in that summer was we deleted a gene, MASH1, and basically when we knocked out this gene, we just wanted to see the effects of this deletion in the mouse. And when we deleted this gene, or we knocked it out, we replaced it with a marker to see the effects of that. I can go more into that, but that's basically what we did. Some of the procedures we did was perfusion, which just involves drawing out the blood from the subjects, from the mice, to study the brains once we put them to sleep, killed them. And we isolated the DNA, we did some genotyping, we amplified the genes via polymerase chain reaction, which a lot of you guys did or will do. and we used NEO as an indicator, and we also performed immunohistochemistry. These are some of the tools. This is a cryostat, and basically we slice some brain tissues there to analyze them when we did immunohistochemistry. And immunohistochemistry just involves staining the tissue samples to see the activity, to just analyze activities of, so basically when we knock out the gene, we insert the brain with a marker, and then we just slice the brain and look at it under a microscope, one of these, and just analyze the activities there. This is a gel, which many of you guys already know. This is paraformaldehyde. This is to preserve the tissue sample. And this is where you perform the PCR. This is polymerase chain reaction. Basically, you just want to amplify a fragment of DNA that you're interested in. So basically you just need one template, one strand, and you end up with what's it called? Exponential. It increases exponential, the amount that you have. So you have a whole bunch at the end of the day. And it's performed here if you enter the program. And this is mouse perfusion. What we did, we essentially inserted a needle into the heart, into the left atrium. And basically we drew out the blood, and we insert hypernoid saline, and basically we wanted the brain blood-free, so when we did the immunohistochemistry, we wouldn't have false positives, meaning we didn't want blood in the brain, so because the blood could show up as something that is significant, which is not significant at all. This is blood. So we wanted this blood-free. This is the brain that is sliced and stained. Future work, we went to cross heterozygous mice with wild-type mice to get bigger colonies. Basically, the mice we worked with the knockout genes with some wild-type mice, and just examine the progression of obesity in the transgenic mice, the mutated mice, and compare it with the control, which is just wild-type normal mice, which we didn't mutate at all. These are some of the references. Big mistake, Wikipedia. I did this a long time ago. So yeah, that's just a rough overview of what I did that summer.
Can you explain the NEO indicator?
Oh, the NEO? Well, when you knock out it. Oh, can I explain the NEO indicator? From what I remember, when we knocked out the MASH1 gene, we needed to insert something in its place. So we used the NEO to see that we effectively did knock out the MASH1 the gene. So that's replace it with something.